Welcome. In this session, I'm going to discuss about the chromosomal theory of inheritance, and then later I'll also discuss about linkage and recombination. Now, before I discuss chromosomal theory of inheritance, I would like to tell a few points on why Mendel's work was remained unrecognized for a long period of time, that from 1865 to 1900. Okay, so Mendel's work was published in the year 1865, but for some reasons it was not recognized by anyone, okay, until 1900. So in 1900, three scientists rediscovered rediscovered Mendel laws. Okay, and I think I told you already about these three scientists in the first session. So they are Hugo de Vries. So this person is from Holland, and another person, Karl Correns, is from Germany, and another person from Erich von Techmark from Austria. So these three scientists, they did, okay, experiments independently. So they did experiments independently and came to, okay, same conclusions as that of Mendel. So that's what we say. So these three scientists rediscovered, okay, Mendelian laws. Now, why from 1865 to 1900 Mendel's work remained unrecognized? So first we'll focus on those, okay, points. So let's see reasons for Mendel's work unrecognition. So reasons for Mendel's work unrecognition. So here the first point is communication in those days was not easy. So in those days was not easy because, okay, there were no telephones, okay, and any other media except the newspapers, which were also very less during 1865. Okay, so hence his work was not widely publicized. So here the communication in those days was not easy, and because of this, so due to this, okay, his work was not widely publicized. Work was not widely publicized. So this is one reason why Mendel's work remained unrecognized for a very long time. Then the next point is concept of factors. So his concept of factors as a stable and discrete units that control, so that control expression of traits, that control expression of traits. So he told that there are, okay, certain things, what he called in his words factors, and he told that these factors are stable and they are discrete units, and they control the expression of traits. And this point was not accepted by his contemporaries. Okay, so this was not accepted by his contemporaries, means the scientists of his time. So no one agreed with Mendel. So that saying as factors as a stable and discrete units. So that is another reason for Mendel's work unrecognition. Then another one is his approach of using mathematics. So his approach of using mathematics, okay, to explain biological phenomena, to explain biological phenomena was completely new. So it was completely new. Okay, biological phenomena was totally new, and okay, and was not accepted, okay, by the biologists of his time, okay, and was not accepted by the biologists of his time. So they did not agree using the mathematics. Until then, no one in biology used mathematics. So he's the first person to explain biological phenomena, okay, by using mathematics. Okay, so his approach of using mathematics was completely new, and okay, no biologist of his time agreed with Mendel. So this is another reason for his work going unrecognized. Then he could not provide a physical proof. So he could not provide a physical proof for existence of factors. So he told that factors are present and they are being transmitted from parents to offspring, and they are stable and discrete, but he was not able to, okay, provide any physical proof, means where they are present, how and what they are made up of. So all those things he was not able to explain. So I already told you in previous sessions that okay, during his time there is no word called gene in the dictionary of biology. Okay, so and no one knows what these genes, what they are made up of, all, okay, all those things. And Mendel, okay, also was not able to provide any physical proof for the existence of factors. Okay, so these are some reasons, okay, for Mendel's work going unrecognized. Right now, by the okay time 1902, so there were a lot of developments in microscopy. So there were significant development in microscopes. They developed microscopes, and scientists, they started studying the cell divisions under the microscopes, and they were able to observe, okay, some colored bodies. That's what we call them, chromosomes. So they started seeing these chromosomes, how they are moving. Okay, so all this movement they started seeing by 1902. Okay, so here, coming to chromosomal theory of inheritance. So chromosomal theory of inheritance. So this theory was proposed by Walter Sutton and Theodore Boveri. So it was proposed by two scientists, Walter Sutton, who is an American genetist and physician, whereas Theodore Boveri is a German biologist. So these two persons proposed the chromosomal theory of inheritance, okay, in the year 1902. Okay, of course, during the Mendel's time there were no good microscopes, but by 1902, Okay, microscopes, there was a development in the microscopes, and the scientists started seeing, okay, these colored bodies called chromosomes under the microscopes. Okay, right. So these chromosomes, okay, where chroma, which means okay, color, and soma, which means body. So we call them as colored bodies because these chromosomes, they take up stain. So when we apply stains, they take up and appear, okay, as colored bodies. So because of that, they called, okay, as of course chromosomes, okay, colored bodies. And here we know already in the first year we have seen about the chromosomes, and also types of chromosomes based on the position of centromere also we have seen. So this is a chromosome. Okay, right. So this is actually a unreplicated chromosome, is an unreplicated chromosome. Okay, right. And if it gets replicated, we will be seeing two chromatids. So we'll see two chromatids. Right, so you can see here, so this is a replicated chromosome. So in this we find, okay, two chromatids. So these two are, okay, sister chromatids. So they are chromatids, okay, two chromatids. In case of a replicated chromosome, and replicated one will be having only, okay, one chromatid. Okay, of course, when we say here, if it is only present, we usually call it a chromosome. Okay, and here, if you ask how many chromosomes are present here, and how many here, so this is only one chromosome. So this is one chromosome. Okay, this is also one chromosome, also one chromosome, right? And I told you that Walter Sutton and Theodore Boveri, both of them, they started seeing these chromosomes, how they are getting separated during the cell divisions. We know there are two types of cell division, mitosis and meiosis. Okay, so the mitosis, which is equational division. So mitosis is a equational division, whereas meiosis is a reductional division. So this meiosis is a very important, and this one takes place during, okay, formation of gametes. So occurs during formation of gametes, okay, in a diploid organism, in a diploid organism. So it occurs during formation of gametes in a diploid organism. And of course, the meiosis is the basis for, okay, variations or recombinations. Okay, that later I will discuss about. So meiosis, it occurs during formation of gametes in a diploid organism, and it's a reductional division. So just briefly give you an idea why the equational, okay, and reductional division. So if I take the a parent cell with 8 chromosomes, so this is the parent cell with 8 chromosomes, that is 2n is equal to 8. Now when it undergoes the mitosis, we know there will be two daughter cells formed. Both the daughter cells will have same number of chromosomes. So that is the reason why it's called equational division. And if we take in case of meiosis, So this is the parent cell, and it also has say 8 chromosomes. And now the meiosis 1, so it will be reducing the chromosomal number. Okay, I'm sorry, reducing the chromosomal number to half of its parent. That will be 4, will be 4. Okay, now the meiosis 2, so we'll have total 4 cells, each one with the same number of chromosomes. We know at the end of meiosis 4 gametes are formed. So 4 gametes are formed. Okay, so you can see in case of meiosis, the chromosomal number is reduced exactly half to that of its parent, and that is the reason, okay, why it is called meiosis as reductional division. Okay, now let's see about this chromosomal theory of inheritance. So here I told you Walter Sutton and Theodore Boveri, both of them, they observed this movement of chromosomes, and okay, the behavior of chromosomes was parallel. So they came to know that the behavior of chromosomes was parallel to the behavior of the factors told by Mendel. Okay, so here, so according to this theory, the behavior of chromosomes is parallel to the behavior of factors. Of course, those factors now we are calling as genes. Okay, right. And we also have come to know that these factors are present on chromosomes. Okay, so say suppose this is a location where you find, okay, the character that is tall, right, and somewhere here, okay, for round color. Okay, just I'm giving an example that what we have seen earlier. Okay, so this is the seed shape for which round, okay, and for this tallness, okay, this is how the factors are present on chromosomes. So here I simply say that the factors are present on chromosomes. So this is what the physical proof that was provided by Walter Sutton and Theodore Boveri. So when Mendel was asked to provide the physical proof, he was unable to provide. So because in his time he do not know about all these things, right? So by 1902, we have come to know whatever that factors told by Mendel. So those factors are present on chromosomes. Okay, right. So how we have come to know is that the behavior of these genes is parallel to the behavior of chromosomes. So how the chromosomes are moving during the meiosis, okay, the same pattern is followed by the factors. Okay, so with that, okay, they proposed this chromosomal, okay, theory of inheritance. Next, we know that chromosomes occur in pairs, as well as the factors also occur in pairs. Okay, so chromosomes occur in pairs and factors also occur in pairs. And two alleles of a gene pair that will be located on homologous okay, sites on homologous chromosomes. So this is I'll mention here. Okay, say suppose these are homologous chromosomes. Okay, we know that in case of homologous chromosomes, okay, so they are actually the chromosomes who have similar size, shape, and structure are said to be homologous chromosomes. Say this is a homologous pair of chromosomes. So here in a homologous pair of chromosomes, okay, one chromosome will be from, okay, one of the parents, or that is either male, okay, and another one, I mean, one will be from male and one will be from, okay, female. So let's say if this one is from the male parent, this is a chromosome from, okay, male parent. So this is what I call it paternal chromosome, and this one will be from the, okay, female parent, and I call this as maternal chromosome. So one is a paternal chromosome that comes from the father, and one will be maternal chromosome. So every one of us have 46 chromosomes. Out of 46 chromosomes, 23 chromosomes have come from father. So means there are 23 paternal chromosomes, and the remaining 23 have come from the mother, and they are called maternal chromosomes. Okay, so the first chromosome of paternal side will pair with the first chromosome of maternal side. So now those two are said to be homologous pair. Okay, okay, say suppose here this is the first chromosome of the paternal side, and this is the first chromosome of maternal side, and second one with the second one, third one with the third one. Never it will be, okay, the third one with the first one, okay, or first one with the seventh one. Okay, so they are not homologous chromosomes, right? So here, okay, so this is the paternal chromosome, first one, and the maternal side, the first one, and you can see here the genes, and its pairs will be present on homologous sites. Means, so here is it's a homologous site, and say here it has small t, and in this case it is small r. You can take a capital T, okay, here. So if it is the case, then it will be tau. In this case also it will be tau. So if we take here in both the cases, so the, I think you now you understood that why I used to take double T's, okay, double R, double Y, okay, when I used to take parents. Okay, so this is how they are present. So if it is the case, then this will be, okay, a plant with tall, so that will be growing tall, and its seeds will be round in shape, because I told that round is dominant. Okay, right? So these are said to be homologous sites. So anyway, we have understood that the factors that were told by Mendel, we have come to know that they are present on chromosomes. That is a the physical, so that's a physical proof for the existence of factors, right? So one point I told, chromosomes occur in pairs, and also factors occur in pairs. That's, I'll mention. So similarities between chromosomes and factors. That's genes. So what are the similarities exist between chromosomes and factors? So one point I told that both occur in pairs. So that's one similarity. Another one that, okay, both segregate at the time of gamete formation. So both, okay, segregate at the time of gamete formation. Means they get separate at the time of gamete formation. So when I used to tell that capital T small t, okay, this parent while forming the gametes, we know that okay, the capital T entered into one gamete and the small t enters into another gamete, so that both segregate at the time of gamete formation. So here I'm telling about the factors which are getting separated at the time of gamete formation. At the same time, the chromosomes will also separate at the time of gamete formation. That part I will shortly discuss. So this is another similarity. Then independent pairs. So then independent pairs segregate independently. So independent pairs segregate, okay, independently of each other, okay. So of course just now I have shown that independent pairs will get segregate independently of each other. So we will see these points by taking the meiosis, okay. So here I'm coming to this point again. Of course this is unreplicated chromosomes. If I show a replicated, okay, chromosome, that's the centromere. Okay, so now these are replicated chromosomes. Now if it is the case on this chromatid also you find here small t, here also you will find capital R, okay, if it is the case. Then here you will find again, okay, small t, and here you will find small r. Okay, I'm just taking, okay, few alleles on the chromosome. So now let's see, okay, how they get separated, okay, in the process of meiosis. I'm going to take a diploid cell whose 2n is equal to 4. So here I'm going to mention, okay, so there are two, okay, paternal chromosomes. Of course whenever we see 2n is equal to 4, which means in this case 2 will be, okay, paternal chromosomes and 2 will be maternal chromosomes. Okay, so here I just mentioned two paternal and two maternal chromosomes, and we know, okay, the crossing over, okay, takes place between, right, maternal and paternal chromosomes. Okay, so I hope we know this is a sister chromatid. Okay, these two are sister chromatids, these two are sister chromatids, and but these two will become, okay, non-sister chromatids. So they are non-sister chromatids, and the crossing over will take place between non-sister chromatids of homologous pair of chromosomes. Uh, Anyway, just I'm going to mention here, okay, small t and r. Okay, so on this we have seen, okay, or I'll just mention, we'll take only one. So on this capital T it's present, and say on this we have capital Y, okay, capital Y, and here we have is, okay, small t, and here I'm going to take a small y. So now during the metaphase 1, so in the next stage will be the metaphase 1. In the metaphase 1, the chromosomes will align, okay, on the equatorial plate, we know it, okay. So how they are going to align? So they are going to align randomly and in two rows. So in case of metaphase 1, chromosomes, okay, align on equatorial plate randomly and in two rows. If it is mitosis, then we will see the chromosomes aligning only in one row. But there is a speciality in case of metaphase 1. Okay, of course, metaphase 2, you will see the chromosomes aligning in only one row, but we will see here they align in two rows and they will be randomly. Okay, which means we should not take like paternal chromosomes will align on the top and maternal on the bottom. No, they randomly align. So here I'll just show. So this is okay. Say suppose these are maternal chromosomes and these are paternal chromosomes. Okay, maternal or paternal chromosomes. So you can see here they arrange, okay, randomly. Okay, and here, so this is the equatorial plate, right? And we can see here there are, okay, on this here is a capital T, so here is, okay, small t, and here we have is capital Y, and here we have is small y. Now, so at the end of this, we know we are going to get two daughter cells. We're going to get, okay, two daughter cells, and okay, two daughter cells. So here in metaphase 1, okay, it's the chromosomes separate. So chromosomes get separated. So you can see here the chromosomes got separated. Okay, right. So we can see here this is capital Y, okay, and here is small y. So you can see the alleles, they got reduced to half, and here also this will be, okay, capital T, and in this case it is small t. So after meiosis 1, okay, this is the result. So in metaphase, okay, the chromosomes will align in two rows and align randomly, and then during anaphase, okay, it's the chromosomes that they get separated. Okay, and this is how at the end of the meiosis the cells are seen. Now these cells again, they undergo the meiosis 2. Okay, so in that case we'll see here the chromosomes aligning only in one plane here, right? So we'll see capital Y, and this is small t, in this case capital T, and this is small y. Okay, now, okay, anaphase. So later in the anaphase, okay, here chromatids will get separated. So this is the metaphase 2. This is the metaphase 2. So here in anaphase, what happens is okay, chromatids will get separated. So here anaphase 2 chromatids get separated. Okay, so we can see here this will result in the formation of two daughter cells, and this also results in the formation of two daughter cells. So here, okay, so into one cell one chromatid, and this is another chromatid. Okay, and here also same. Yes, so you will be seeing all something like this. Okay, so here say this is having capital Y, and here it is, okay, small t. Of course in this case also it will be, okay, small t here, and here it will be capital Y. So in this case it's a small y here and small y here, and capital T, capital T. So these are the gametes which are formed. So all are haploid, right? So we know, I told you that 2n is equal to 4, which means here n will become, okay, 2. You can see, and how the chromosomes they got separated. And when chromosomes get separated, the, okay, genes present on them or the factors present on them also get separated. Okay, so this is about the chromosomal theory of inheritance. Now the experimental proof for this theory was given by another scientist named T.H. Morgan, so called Thomas Hunt Morgan. Okay, so experimental verification. So experimental verification for this theory was given by Thomas Hunt Morgan. So was given by Thomas Hunt Morgan. In short, we can call, okay, T.H. Morgan, and he did experiments on, okay, a fruit fly. So Thomas Hunt Morgan did experiments on fruit fly whose scientific name is Drosophila melanogaster. So it's called the fruit fly, scientific name Drosophila melanogaster. So here there are certain reasons for choosing this fruit fly for his experiments. So let's see those reasons for choosing Drosophila. So reasons for choosing Drosophila by T.H. Morgan, okay, and his colleagues, they have done this experimental verification of the chromosomal theory of inheritance. So let's see, so what are those reasons for choosing Drosophila? One, they can be grown on simple synthetic medium in the laboratory. So they can be grown on simple synthetic medium in the laboratory. So that is one reason for choosing Drosophila. It means that it is very easy to grow them in the laboratory. Then next one, they complete their life cycle within 2 weeks. They complete their life cycle within 2 weeks, which means they grow, reproduce and die, okay, within 2 weeks. So that's what they complete their life cycle within 2 weeks. Then a single mating a single mating can produce large number of offsprings. So it can produce large number of offspring. So each one of it may produce 3000 eggs. A large number of offspring can be produced, a single mating. So that is the one reason. So we know the more the number, the less is the chance of error. Okay. The next one is there were a clear differentiation of sexes. Okay. Okay. There. Okay. So there are clear differentiation of sexes, so we can easily come to know which one is a male, which one is a female. Okay, so clear cut differentiation exists between, okay, male and female. So that is a clear differentiation of sexes and also the characters. Okay, so we require, okay, contrasting characters, how we have seen in case of pea plant. So here also we find a lot many of them, okay, in Drosophila. So these are some reasons why Thomas Hunt, Morgan and his colleagues have chosen this Drosophila for their experiment. Of course they did complete it, and they also proved the theory, chromosomal theory of inheritance. Like, so that's all about this theory. The next one we shall see about linkage and recombination. So the next one, linkage and recombination. Okay, so okay, first I'll talk about recombination. So what actually is this recombination? So we know that there will be a crossing over that will be seen between, okay, non-sister chromatids of a homologous pair. Okay, say suppose a gene from this point, so it, okay, gets over here and this one comes over here. So like this, at different locations, I'm just mentioning, there will be, okay, crossing over. So crossing over. So this crossing over results in recombination, means, okay, on this there will be genes which are present in linear fashion. So the genes will be present in, okay, linear fashion. So say here I'm taking, okay, again, so from here to here there is one gene, from here to here another gene. So like this, one after the other. Of course there may be regions where we don't find anything. Of course there will be nitrogen bases and they don't code for anything, so we will not carry them, but the genes are seen in a linear fashion. So here genes arranged, okay, in a linear fashion on chromosomes, linear fashion on chromosomes. Okay, now, okay, so this gene is been replaced with something else. So here earlier, say suppose it is X, but now it became Y. Okay, so say I'll take A, B, C, D, and after combination, so here it has come X, and some here it has come, okay, M. So you can see this combination has been changed. So that's what it's called, recombination. So crossing over resulting in recombination, and this recombination is the one which is responsible for production of, okay, variations, means differences in the offsprings. Okay, right. Now what is this linkage? So let's see here, when two genes are present very close to one another, okay, say, so here we have a gene and here is another gene. Okay, so say gene X and this is, okay, gene Y. So I'll take another one. Okay, so this is gene Z, and I'll take, so this is gene A. Say suppose these are the genes, okay, located on two chromosomes. So you can see here this gene X and gene 2 are present very close to each other. So located close to each other. So when they are located very close to each other, the crossing over will be less. So here the crossing over to take place, there is a less chance. Okay, so hence these two genes will not get separated, so they remain together. And when I compare this gene X with gene Z, so you can see there is a lot of distance. So easily the gene Z and gene X can undergo crossing over. Okay, so when the two genes are located very close to each other, okay, so there is, okay, less chance of crossing over. So there is a less chance of crossing over. Okay, so that's what I'm telling about gene X and Y. So when they are not undergoing the crossing over, the genes will remain together. Okay, so no crossing over will result in genes staying together. The genes will stay together here. Okay, and that's what in case of gene X and gene Y. So but when we compare gene X with the gene Z, or gene Y with the gene Z, or you can take between gene Z and gene A, okay, so crossing over can take place. So these two genes, now I can say, if they do not undergo crossing over and inherit together, then these two genes I say as linked genes. So I call, okay, these two genes as linked genes. But the gene X and gene Z, so gene X and gene Z, they are not linked. And even the gene, okay, X and gene Y, they are also not linked. And if I compare gene Z and I'm sorry, here gene X and gene Y and gene Z, they are also not linked. Okay, so this one and this one. Then gene Z and gene A, so these are also not linked. Only the gene X and gene Y are linked genes. So why we call them linked genes? Because they are escaping the crossing over, and they are staying together and getting inherited together. So such genes are said to be linked genes, and the phenomenon is called linkage. Okay, so right, I hope it's clear. Now I'll just mention the linkage definition. So this will be the tendency of genes. So tendency of genes located on same chromosome. So tendency of genes located on, okay, same chromosome proximal to each other, okay, tend to escape crossing over and get inherited together. So tendency of genes located on same chromosome proximal to each other, means very close to each other, tend to escape crossing over and get to inherit together. So that's we call it linkage. So here I have given an example of. So when you compare this gene and this on different chromosomes, they are not linked. So for a linkage to be seen, the chromosome should be present on the same chromosome, and they should be present very close to one another. Okay, so with this I will end this session, and in the next session we will detailedly understand by taking an example, especially the Drosophila melanogaster, and see how the linkage occurs.